Today's session is entitled Sustaining Biodiversity. A consequence of the large-scale forest destruction is a remarkable loss of species diversity. Numerous plant and animal species have either become extinct or are threatened with extinction. As a consequence, research on biological diversity has intensified. In 2005, Hooper and co-authors published a much-cited review. The authors were certain that more species are needed to ensure a stable supply of ecosystem goods and services. They had high confidence that practices that maintain a diversity of organisms will help preserve a range of management options. And they proposed that long-term experiments are needed to assess temporal stability. Quite remarkable at that time, because everybody had been talking about diversity, but temporal stability seems to be a new era of forest research emerging just now. We have included that remarkable paper in the accompanying material. And what I like about this review is they are certain about something, they have high confidence about something, and they don't know much about something. So they recommend that further research is needed in an area that they consider to be important and that they don't know much about. This is my framework. The session is divided into 3 parts. The first part is entitled Perspectives. We will start with Whitaker's hierarchy and try to translate his concepts developed during the 1960s into today's world of anthromes. The title of the second part is Toolbox. I will present tools for assessing biodiversity, for developing a species-area relation and a species abundance distribution, and methods for monitoring the sustainability of a harvest event with reference to biodiversity. The third part, entitled Future Research, deals briefly with plant traits and trait complementarity. I hope that when we finish today, you will had a fresh look at biodiversity and that you will be able to use the methods in the toolbox in your own time. Robert Harding Whitaker was born in 1920. He died in 1980 at the age of 60 years. From the earliest days of his career, Whitaker challenged major paradigms in ecology and evolutionary biology. He was fired from his first academic job at Washington State University and struggled for 6 years to have his dissertation published. Robert Harding Whitaker studied vegetation patterns in the United States, in Israel, in Australia, and South Africa. He challenged Clemens' regional climax theory and emphasized the individualistic nature of species distributions. He transformed a discipline which had become transfixed with categorization into one which could embrace the complexity of variation in landscapes. Whitaker's alpha, beta, and gamma diversity have become general textbook knowledge, and we had one session that dealt specifically with beta diversity 2 weeks ago, presented by Professor Zhang Chunyu and his students. Alpha and gamma diversity share the same characteristics and are differentiated only by the scale at which they are applied. Biodiversity is the one which receives the most contradictory comments regarding its usefulness. And the question is, would it be possible to use Whitaker's hierarchy to get to grips with today's reality? What is a landscape? Where does it end? Is the landscape a continent? Is it a country or perhaps a farm? The classic biomes paradigm says that the biosphere is shaped primarily by the biophysical environment. Ellis and Ramakati have shown that the major factors shaping the world's ecosystems are population density, affluence, and available technology. Most forested landscapes are not biomes, but anthromes. Anthrom maps now appear in numerous textbooks and even in the National Geographic World Atlas. Introduced or invasive exotics, and the human footprint seems to be everywhere, even in strictly protected areas. We've just completed a study based on 3 tree map plots maintained by the Environmental Management and Policy Research Institute in Bangalore, in South India. The names are Doresanipalya, Thale Woodhouse, and Bugurikalu forest. These 2 study areas are located in the strictly protected Bannerghatta National Park, south of the city of Bangalore, or also known now officially as Bengaluru. And the Doresanipalya forest is located in the middle of a built-up area within the city of Bengaluru. All species were identified by experienced field botanists. Ugurikalu has 76 tree, shrub, and climber species in 35 families. Doresanipalya has also a high number, 46, and Thale Woodhouse, 69. And you can see that in Ugurikalu, 2 species are invasive, although they are protected, 10 planted or invasive, and in Thale Woodhouse, 1. So some permanent study areas in the Peruvian floodplain forests maintained by Dr. Juan Rommel Baluarte Vasquez have more than 130 species per 1 hectare. Such study areas are very rare and very precious. They are invaluable for biodiversity research. And I'm very thankful to be able to participate in the research about these very interesting study areas in the dry tropical forests of South India. Few primary forests are left. These few remaining natural relics are not only cherished for their beauty, but especially for their ability to develop stable communities. They include a variety of tree species with individual traits that allow some of them to survive periods of drought, and others to utilize a favorable climate for rapid growth. It is reasonable to assume that stable natural communities are those in which the traits of the individuals are diverse and complementary. Industrial plantations can be seen as the other side of the coin. Many planted forests are highly productive ecosystems managed with optimum rotations, optimum thinning regimes, pruning regimes, and sophisticated site clone matching. I remember when Dr. Brandão from Brazil visited us in South Africa during the 1980s to tell us about the successful Aracruz plantings in Brazil. Industrial plantations are highly productive, but they often lack long-term stability. How to reconcile these two? How to reconcile high productivity and long-term stability is an objective of many of the run-of-the-mill domesticated forest ecosystems that cover a large part of the land area of the world. Domesticated ecosystems are often represented by a mosaic of spatial units within clear boundaries and often abrupt changes from one unit to the other. These landscapes may include planted forests, uneven age, selectively managed near natural forests, and protected habitats within that in the same landscape. The reality is that most forests are within properties, either state property or communal property or private property. And this map was presented by Jordi Garcia Gonzalo in 2006 in his doctoral thesis in Finland. And it shows a spatial organization of the landscape into units that we normally refer to as compartments. Each compartment has particular attributes, and it is separated from other compartments through very clear boundaries. It's a typical landscape that we find in many parts of the world. We will return in more detail to this topic during the last session on 27 April. So natural forest, stable communities, industrial plantations, high productivity, and many forests that are subdivided into units and thus represent a landscape that is part of a property. And the property may be owned by communities, ejidos, for example, in Mexico, or by the state, or by private ownership. Now let us open today's toolbox. The toolbox includes methods for calculating forest diversity and evenness, for developing a species area relation, SAR, and a species abundance distribution, SAD, and it also includes methods for monitoring a harvest event. The complete data sets of these three plots are available for download. One study areas from Mexico with 10 species, one from northeastern China with 21 tree species, and one from Germany, very close to my home, with 3 different tree species. Dr. Coral Rivas of the University of Durango in Mexico, Dr. Zhao Xiuhai from the Beijing Forestry University, and Dr. Peter Meyer of the Northwest German Forest Research Center have kindly agreed that we may use these 3 data sets in our workshop. Oh, shut it. Oh. Richness refers to the number of species, as I mentioned before, 10, 21, and 3. And diversity is more than richness. You see here the Shannon index formula, where the pi refer to the proportion of the I species and the log pi. This product is summed up. So we get a measure of diversity, and the R code for preparing these plot summaries is available in this document, in a Word document. The code includes a function that makes it easy to prepare these summary tables. I want to go back to the Shannon diversity. The Shannon index is the most common measure of diversity. The index is calculated as the sum of the relative frequencies times the logarithm, as I explained. The variety of diversity indices is bewildering, and many are viewed with suspicion, because most diversity indices are entropies representing the degree of disorder or randomness in the system. Their mathematical behavior does not always correspond to our intuitive concept of diversity. We get a number, we cannot relate that in any way to diversity. However, if we take the exponent of the Shannon diversity, then suddenly it makes sense. I have prepared an example to demonstrate the basic calculations. Imagine 3 forests, A, B, and C. A has 2 species, B and C have 3 species each. They have the same richness, but the relative frequencies are different. I don't know why it doesn't react. My computer is an old machine. I think I'll start again. Sorry about this. Share. Perhaps too many? Are we too many?
Mm-hmm.
No, and I want to share the screen. There's one. Share.
Share, yeah.
Oh, there was a problem with Microsoft Word. I apologize for this. Wait. File, open recent. Yeah, there we are. So this is the one. So I click on it again. So this is a Shannon index. So we get values 0.69, 101, 1.1. These really don't make much sense. The value of C is greater than B because of they have the same number of species, but this forest is more even. The species are more evenly distributed in B than in B. So the Shannon index increases with increasing number of species and with increasing evenness. If we take the exponent, then suddenly it makes sense. We have value, this exponent is also known as hill number. We obtain a value of 2 for forest number 1, 2.74 for this forest, and 3 for that forest. And maximum evenness is attained when we divide the hill number by the number of species. We get complete evenness in the first, in the A forest. We get less than evenness in the B forest, and complete evenness again in the sea forest. So I think this is common knowledge. We need not dwell on this very much. But perhaps it might be interesting for you to have a look at 7 Diversity DocX, this document, which includes the code for our 3 areas, the 3 study areas that are part of this workshop. So we have 10 species, 21 species, and 3 species. The Shannon indices calculated, they have not much meaning. Then the hill numbers calculated, and then the evenness is the highest in the German plot and the lowest in the plot from northeastern China, 0.53, because the species are very unevenly distributed. So you can try these methods and use them with your own data. Now let's have a look at the spatial diversity. Any discussion on diversity would be incomplete without a reference to spatial effects. This is the German plot, and you see 3 circular plots with radius 10 meters in that plot. And we can study the diversity in a small scale, close range neighborhood. This would, by just dividing the number of species by the number of trees. You see, this is black, 3 blues. So there are 4 trees and 2 different species. So 2/4 would give us some value of diversity. Or in this particular case, there is 1 black and 3, the same value. And here we have 2 species again and 6 individuals. So 2/6 or 2/4, and sometimes. So if we allocate these circles, many of these circles, into this plot, then we get a measure of the distribution of diversities, a close range spatial diversities. And I've done this, and I think, yeah, you can also do this with your own data. We call this mingling, the proportion, the ratio between the number of species and the number of individuals in close range neighborhoods. So the mingling, the mean mingling in the Mexican plot would be 0.3, in the China plot 0.3, and the German plot 0.34. The mingling value higher in the German plot, although it only has 3 species in contrast with 21 and 10 species. Much higher diversity in the Mexican and China plot, and low diversity, low species diversity, but a higher spatial mingling. You may remember 3 weeks ago during our session on structure, density and structure, I talked about individual mingling. That means we, instead of putting in circles, we take we consider specific trees, specific individuals, for example, all the quercus, the red ones, and look at the distribution around an individual. In this case, we call this group mingling. The group mingling refers to a fixed radius circular plot where the number of individuals may be variables, not fixed, as in the individual mingling. Well, a very big topic. I just want to touch on the possibility to look at diversity in a spatial context. The value of a diversity index is meaningless unless it is expressed per unit area. 10 species on 1 hectare is not the same as 10 species on 3 hectares. We are evaluating a large number of manuscripts each year in forest ecosystems. Every now and again, Shannon diversities are presented in studies on biodiversity. Such results are meaningless unless there is a reference to the area sampled. The basal area of a forest is a meaningful density measure only if it is expressed per unit of area. For example, basal area per hectare, then it makes sense. The same applies to biodiversity. The differences in microsite conditions generally increase with increasing area. Accordingly, the variety of species that can be supported increases with increasing contiguous area. The relationship between the contiguous area and the number of species is known as a species-area relation. This relation has been modeled using asymptotic function, like this one, power function, this one, and the so-called mono function, monot. Mono was the French physicist that published this function. When we take many random samples with increasing plot radius, circular samples, increasing radius, then we get this relationship between the area sampled and the number of species. We can then fit these 3 functions. And when we fit the functions, in this particular case, green is the asymptotic function, this one, the green line. Blue is the power function, and red is the mono function. You can do it yourself if you look at the R code. We have published a study in 2007 that confirmed a suspicion by Hubble and others that the tree species area relationship can be derived from prior knowledge of the total number of species in a particular region. The studies is that study is also in the download folder, so you can look at it at your own time. It would go too far now to explain it in detail, but in question time, I can try and explain it in more detail. We found the mono function to be especially flexible and suitable for small plots as well as large sample plots. In a small plot, for example, very small plot, we are never sure about the maximum number of species. So the power function sometimes works very well in small plots. In very large plots, where the asymptote, where we're sure that we've reached the maximum number of species, the asymptotic function works quite well. But the mono function is flexible in both small plots and in large plots. Here you see the fit is quite good in the Mexican plot, also in the China plot, and also in the German plot, although there are only 3 species. The second part in the toolbox concerns the species abundance distribution. This picture was taken by Dr. Juan Rommel Baluarte Vázquez from Peru, and it shows typical landscapes in the Peruvian floodplains. The number of species in this particular plot, T7, was 139 on 1 hectare. So the curve here shows the abundance rank. Eschweilia parvifolia is the most abundant species, has the high, on the log scale, a very high abundance, followed by another one, then less and less and less, and many species that have that are represented by only one or two individuals. So typical, they call it the hollow-shaped species abundance distribution, which describes the relationship between the number of species in the field study and the function of their observed abundances. A popular model that we can use to fit such data is the log normal distribution. I found the Weibull distribution very suitable, especially for this kind of shape. The histogram at right shows the number of species on the x-axis and the number of individuals on the y-axis. So how to interpret this? We need to think a little bit how to interpret this. There are 60 species that are represented by very few individuals, one or two individuals. 60 of the 139 species are very few individuals. And there is one or two species are represented by a very high number of individuals. Again, the typical abundance distribution that we find in species-rich forests. Now, I've done this again to allow you to do it yourself. These are the three species abundance distributions for our Mexican, China, and Germany plot. And you see typical curve shapes in the Mexican and also more pronounced even in the China plot. And here we only have three species, of which two are quite abundant and only one is less abundant. You are welcome to try the code in the download material in your own time. The shapes of these curves are very sensitive to scale. The paper by Tanling Zhao shows this very nicely. When you change the scale, normally with a smaller scale, at smaller scale, normally the curve shape becomes more hollow. And it would become more like this if the scale is less. And if it's bigger, it will become more hump-shaped. The third part of the toolbox deals with harvest events. Monitoring harvest events is a basic requirement of sustainable forest use. The removal of a tree represents a sudden, dramatic disturbance with many effects on the microclimate, the soil chemistry, the habitat functions, on the regeneration, and the ground flora. A harvest event modifies the structure and biology of the forest and has long-term implications for the ecological and economic sustainability. We can characterize a harvest event by weight or severity. The reduction of the density may be expressed by the proportion of the basal area removed. Or we can also characterize the harvest event by size selectivity. If we divide the relative number of trees removed over the relative basal area of trees removed, we get this ratio Rn over Rg. Rg is this one. Then we get the so-called Ng ratio. The higher the value of this ratio is, the more selective is the harvest on, the higher is the preference on selecting small trees. And the lower that value, the selectivity is greater on the big trees. We can also define a harvest event by species selectivity. If we divide the removed individuals of one particular species over the total number of individuals of that species before the harvest. And we can also evaluate the effects on spatial mingling or on dominance within close range neighborhoods. I will just show one or two examples. Imagine two species forest. The RG ratio and the NG ratio, the type and the severity can be identified for each species A individually. A relatively small proportion of the basal area is removed, and most of the individuals of this species are the small ones, or the selectivity in species B would be a large part of that species has been removed in this harvest event, whereas only the big trees, mostly the big trees, have been removed. 40% of basal area, and the NG ratio is 0.5, which means the big trees have been preferred. Selectivity can be expressed, as I said, using species selection preferences. This is a 11 hectare, 10.9, almost 11 hectare plot maintained by Professor Zhao Xiuhai. These are the trees before the harvest, and these are the removed trees. Not one Pinus coriensis tree was removed. There were 339 Pinus coriensis trees before the harvest. Not one was removed, so there's no preference of that. In contrast, Betula platyphylla, there were 96 trees of all the 27,000 trees, only 96 trees, but 7 of those 96 trees were removed. So very high preference and index, almost 7%. In Acer mono, which had 2,455 trees before the harvest, 98 of these were removed, so a preference index of 0.4. RG values and NG ratios are also given, and it seems that only the big trees were harvested in this particular plot. We have published this study some years ago, and I forgot, I need to put the reference here. I forgot to put the reference. I think it is part of the download material. If not, then I will include it and ask Huanzhen to include that. We have found harvest event teaching to be a most interesting part of the teaching of forestry students, because it brings together all the different disciplines. We selected a harvest event area, then the students would mark the trees, and later on they would discuss a particular selection in different plots, and present were all the colleagues from the different wood science department, from the economics department, biologists and ecologists were present. So it was a wonderful teaching exercise that brought together different colleagues with their specialized knowledge. A review on biodiversity would be incomplete without a reference to traits. Traits are inherent attributes of plant species. Examples of traits are wood density, maximum tree height, seed mass, growth form. Growth form refers to tree, shrub, or climber, or the lifespan of a tree. Dr. Jens Katke from the Max Planck Institute in Jena, in Germany, maintains a database with about 12 million trait records for a large number of plant species. Important response traits are plant tolerance to frost, to drought, or to shading. As Dr. Katke explained in this diagram, traits may change according to tree level attributes. Oak trees are shade tolerant only at a very young age. Traits may be modified also by site. Maximum tree height may be severely reduced on wind-swept sites near the coast. Response traits also determine the capacity of a tree species to establish itself and survive in harsh environments, at high altitudes, in deserts or salty marshes. If you look at Hauman Hui's publication, which is included in the download material, we have published a study on traits and trait similarity and dissimilarity between different forests. So congratulations to Dr. Jens Katke. I think this work is extremely important because traits in a community of trees can be complementary, and trait complementarity may become a very important aspect of forest ecosystems research. There's growing consensus that functional diversity rather than species numbers determines ecosystem functioning and stability. Functional diversity refers to the value and range of species traits. Species diversity is not always an adequate surrogate for functional diversity, and we can expect a surge in trait-based research during the coming years. Different species may have very similar response traits. The similar traits indicate different resource strategies. Sustaining means to ensure the long-term stability of domesticated ecosystems, and trait complementarity may be more important for long-term stability than diversity. These questions require new research efforts. Whitaker Revisited. The topic of next week's presentation is Stanley delineation. The presenter is Professor Timo Pukala from the University of Eastern Finland. Timo is a leader in research that deals with sustaining forest ecosystems. The session promises to be highly interesting. This last slide presents a glimpse of the Skola forest in the Ukrainian Beskids, where Dr. Mykola Korol is doing his research. Thank you very much for listening.
Thank you, Klaus.
That was my...
You? In this session, I will try to organize the question. Yes, good morning, Klaus. And you can write in the chat or put your hand here, not in the system because it's difficult for me to see the hands. Then you can ask directly or in the chat. Yes.
I'll leave the PowerPoint on. So if you have questions to a particular slide, then we can go to that slide, okay? I can't see the chat. Where can I?
No.
You can, hi, Carlo.
Yeah.
Yeah, I think Dave is here.
David Drew?
Yeah.
Hi, David. Where are you?
Klaus, I'm here, but I'm happy for Alicia to chair the session. It's fine with me.
Why you're in the field? How you manage that?
No, I'm not in the field. It's just a background.
It's good.
Oh, yeah.
Alicia, would you like to chair or would you like me to take over?
Alicia, let Dave do it. There you go. It doesn't matter. Both of you are welcome. You do?
There are no questions. I have a question. Oh, Oscar Pierce from Universidad Austral de Chile said, in relation to long-term trials, there is a minimum data structure to consider for sustainable forest plantation management objectives.
I can't see the question. Wait. There is a minimum. A minimum. And minimum what?
Data structure.
The data structure. Minimum data structure to
consider the sustainable forest plantation management objectives.
Oh, wow. I'm thinking a minimum data sector. The question, minimum data question, has always been cropping up every now and again, but I'm trying to figure out what the minimum data requirement is. Had nothing to do with biodiversity, Alicia, or has it to do with biodiversity?
In long-term trials, I think that Oscar Pierce can explain better the question.
Professor, regarding, you hear me? Yeah. Regarding to the forest sustainable management and forest plantations, there is always the question of it is sustainable or not. Yeah, but with this focus today on biodiversity, there is a sum of structure, a minimum structure to research and to help to answer the question about sustainability, a good practice management, especially in forest plantations.
Thank you. Plantations are very important, Oscar. I've worked myself in plantations many years in South Africa. Yeah. But there is always the question of biodiversity with plantations and long-term stability. Long-term stability refers to an ecosystem to persist in times of change of climate in particular. So the two sides of the coin is productivity on the one hand, plantations, well-managed plantations like the ones in Brazil, for example, or in South Africa, also Chile. I believe you have radiata plantations. I've seen your beautiful radiata plantations. Plantations have a very important role. Today's presentation is about biodiversity because it is another aspect is how stable are the ecosystems, how stable are the landscapes where trees, covered by trees. to persist in times of change. And I think this is a very important research question that we as foresters need to address. We need to more to concern ourselves with the biodiversity. And there are examples, study examples from the field of ecology that are very, very helpful and very useful for us. And I thought that if the topic is sustainability, it should not only refer to production, but also should refer to an ecosystem to maintain itself, to be able to persist in times of change.
Thank you.
Well, maybe in the last session, I will try to reconcile these two different types of management. Plantation management, where we have rotations and clear felling, and then a sequence of thinnings and a yield model, with the much more complex management of uneven-aged, multi-species ecosystems that have a high diversity and therefore maybe not so productive, but still able to survive periods of change. And
Lars, there are a couple of other questions here. So the first one that came through on the chat was from Liang Jingjing. And he asks you, what do you think is the most prominent driver of forest biodiversity? Is it climate, soil, anthropogenic, or other factors?
Thank you, Dave. Yes, Jingjing, hi, I can't see you. Thank you very much for your question. And I think considering our presentation on anthroms, if you remember, Jingjing, can you hear me?
Yes, I can hear you.
Ah, there you are. Thanks. And anthroms, I think there is agreement that biomes, the old biomes idea, are more and more replaced by the anthroms. You have worked on anthropogenic biomes yourself. So most areas of the world, even the highly protected ones, are modified by humans. So I think the human force, the anthropogenic force, is a very important one next to environmental, climate and soils and so forth. And then, of course, as we all know, in the tropical regions, we have a high biodiversity, and in the boreal region, a low biodiversity. I think there are other people who are more qualified than me to answer these questions and this particular question. But if you try to, would you like to specify it a little bit more so that I can focus on what is your intention? What do you want to discuss?
Yeah, Klaus, I was just wondering, based on your experience about the plots that you have been involved with, what generally are the factors behind the diversity levels of those sampled area?
Well, the Indian plots, the Peruvian plots, we know it's a very, very high biodiversity. But we know there are many species. We can identify the species. We have qualified botanists can identify the species, but we do not know very much about their complementarity. You know, whether you have deep rooting or shallow rooting species, shade tolerant species, a mix. Some ecosystems are extremely successful. And one of these is the combination between oaks and pines, in the southern United States and Mexico, we have oak pine ecosystems. We have oak pine systems throughout the Eurasian continent, in Korea, up to Korea, from Korea, northeastern China, then right through Europe, oak pine. Why are these two genuses or genera, we say, make ecosystems that are so successful? There must be something that we don't know. Maybe people have studied them, but I think this is just an example to show, I don't know, the diversity in oak pine systems does not seem to be so high. It certainly is high in tropical climates, whether dry or not.